Well, welcome everyone. I'm Mark Schumacher. I'm the director of PARC, and I just wanted to take a very brief moment to remind everyone that the Pain and Addiction Research Center is really a transdisciplinary experiment to bring both the disciplines of pain and addiction together, as you know. Um, as you also know, we offer a monthly seminar series as well as other opportunities for pilot grants. And I just wanted to quickly remind you that next month, in May 27th, we're honored to have Anne-Marie Malfait to join us from Rush University. We'll be talking about processes underlying osteoarthritis and rheumatic disease. Um, Anne Marie is the chair of osteoarthritis research and director of Chicago's Center for Musculoskeletal Pain Research, and we're really looking forward to that. With that, I'm going to turn it over to Dorit, who will be moderating and introduce our speaker. Stop sharing. Okay, Dorit.
So, hi everyone. It is my great honor to introduce George Koob to you. For those of you who do not know George or do not know George's history, he got his PhD at Johns Hopkins, and then he did a postdoc in Cambridge University in England with Leslie Iverson, who I associate with Substance P. He then got position at the Salk and Scripps, and I met him at Scripps when I went to give a talk there. I was very young, and George, you probably don't remember that. You were driving me in your sports car to, I assume, a restaurant, and it was by the ocean in La Jolla, and we are driving sunset, and he goes, with this view, who would want to leave this place? But in 2014, George did leave. As you know, since then, he's been the director of NIAAA, and we're very happy about that. His lab is at the National Institute of Drug Abuse, where he continues his research on addiction. George made some, terms based on his science that everyone uses, including this three stages cycle of addiction, which I'm sure, or I hope he will show here, as well as or together with it, the spiraling down from, in the case of alcohol, recreational use to alcohol dependence. And I have to say that George explained where this spiral came from, and it had something to do with toilet paper. George won many prizes, including the French Legion of Honor, national, and I won't go into trained 100 postdocs and graduate students, etc., etc., etc. He really is the central scientist in our field, in my field, and it is, I won't say anymore, there's a lot to say. It is my great, as I said, honor to have you, George, here on Zoom.
Thank you, Dorit. That was very kind. And yeah, the story was that I indicated that addiction was like a spiral. It was going downhill in a funnel. And some intrepid graduate student put my spiral on a toilet for his poster at the Society for Neuroscience. And that was the end of the spiral. It became a circle after that. So there's always someone who can actually do better than your original intention. But it was kind of funny and fun. Okay, so it's a pleasure to be with you today, and I am looking forward to talking with you about the intersection of hyperalgesia and hyperkatifeia as a driving force for negative reinforcement in addiction. So I don't have to spend much time on this slide. Everybody's pretty much aware of the problem that's involved and why we do what we do in the addiction research field. You know, there's 28 million people with an alcohol use disorder in the United States, 5 million people with an opiate use disorder. You can look at the prodigious number of emergency department visits and hospitalizations for both drugs, and deaths, of course. Alcohol-related deaths are around 180,000 a year and 75,000 for opioids. This is what I'm going to talk about today. I'm going to talk about definitions and a heuristic conceptual framework for understanding negative reinforcement in addiction. This will involve an overview of neurocircuitry of addiction, hyperalgesia, hyperkatifeia, opponent process, and negative reinforcement. I'll be defining most of these terms. And then I'll talk about, but I'm going to make this pretty brief because a lot of it's been covered before. It's, you know, not quite a few review articles. So you get the condensed version of number one. Number two, I'm going to spend some time on the neurobiological basis of hyperkatifeia and hyperalgesia with a focus on between system neuroadaptations. And my laboratory and has contributed, I think, significantly to this area. So I'll be focusing on a lot of the work that we've done in this domain, but again, kind of a synthesis. And then at the end, I'll talk about convergence of brain, pain, and negative emotional neurocircuits. I'll show a little of our new data and what this means for the role of hyperkatifeia in addiction. And I'm going to leave you with a new hypothesis we're starting to look into. Let's get started. So when I define addiction, I define it as a chronically relapsing disorder that is characterized by a compulsion to seek and take drug, loss of control, and limiting intake. You'll find that in every textbook. But the Koob add-on is emergence of a negative emotional state, dysphoria, anxiety, irritability, defined as hyperkatifeia, when access to the drug is prevented. As Dorit said, there's a three-stage cycle with three domains of dysfunction and three main neural circuitry nodes that are associated with this cycle of addiction. I should mention that you enter the cycle of addiction at any point. It's not just in the binge intoxication stage, although that probably predominates. Binge intoxication involves incentive salience and pathological habit dysfunctions, and is mediated largely by basal ganglia and many connections. The withdrawal negative affect stage is represented by the domains of reward deficit and stress surfeit dysfunction, and involves the extended amygdala, which I'll talk more about today because I'm going to focus on that. That's the reason for the red circle. And the preoccupation anticipation stage is largely executive function deficits, craving associated with that impulsivity, and also mediated largely by connections of the frontal cortex. So some definitions. Drug-induced hyperalgesia is defined as a state of diffuse increased pain sensitivity to a stimulus of stable intensity observed during withdrawal from drugs of addiction. The next one, drug-induced hyperkatifeia, is a word I made up. It's defined as the increased intensity of negative emotional, motivational symptoms and signs observed during withdrawal from drugs of addiction. I made it up because I was tired of spending 6 words saying the negative emotional effects, sensitization of negative emotional effects during withdrawal from drugs of addiction. So I slapped together 2 words, katifeia, which means sadness, dejection, and dysphoria in Greek, and hyper. And yes, it was analogous to hyperalgesia, and that's why I did it. This slide is dedicated to Larry Parsons, Lauren Parsons. He was a member of my team at Scripps in our committee on the neurobiology of addiction, and he passed away way too early. But I had borrowed this slide from him, which I thought was very illustrative to describes the progression of the addiction process. And the argument is that reward is, of course, a very important part of addiction, but it fades with tolerance, or hedonic tolerance, as we like to say. And another source of reinforcement manifests itself, which is negative reinforcement, or the decrease in the probability of a response by removing an aversive stimulus. I'll repeat that. The increase in a probability of a response. In other words, taking a drug to remove the aversive consequences of withdrawal, or relief reward, as some people call it. And the other thing I want you to note from this slide is that relapse loops back in a kind of vicious circle. That's the cycle. But it doesn't always loop back to the reward part. It actually probably more likely with chronic addiction loops back to the relief or negative reinforcement component of the progression of the addictive process. So here's a summary of what I just said, put in one kind of nutshell. The nutshell is entitled Allostasis, Opponent Process, Hyperkatifeia, and Negative Reinforcement. So because of opponent process, a greater quantity and more frequent use of the previously rewarding drug are needed to maintain or approach euthymia. And this is called tolerance. When the substance is not used, then negative emotional signs of withdrawal emerge, such as irritability, anxiety, dysphoria, sleep disturbances, and subjective feelings of need. This deficit emotional state, or hyperkatifeia, sensitizes and becomes persistent with repeated use. And we call this now protracted hyperkatifeia. Under this conceptualization, drug intake is compulsively escalated or renewed in relapse via negative reinforcement processes because the drug transiently prevents or relieves the hyperkatifeia. Drug taking for relief ultimately becomes a self-defeating because it worsens and perpetuates the vicious opponent process cycle, creating hedonic allostasis. And that's defined as hedonic stability with an altered set point, and is the basis, in my view, for tolerance. So opioids cause hyperkatifeia and hyperalgesia. This may be self-evident to all of you, but here's actually some data from humans showing correlations of the subjective opioid withdrawal scores, SAWS, and dysphoria from the profile of mood state scores, POMS, in spontaneous withdrawal on the left-hand side, and naloxone infusion, or what we call precipitated withdrawal, in individuals with heroin addiction on the right-hand side. And you can see that there's a straight line, distributed straight line relationship between the subjective opiate withdrawal score and the profile of mood state score. And we know there's also hyperkatifeia from a whole series of studies with human individuals with opiate addiction. In this particular study, in a randomized crossover double-blind trial, saline and heroin were administered to 22 heroin-dependent patients, and 17 healthy control subjects received only placebo. And you can see that the healthy controls have low levels of anxiety and activation of the hypothalamic pituitary adrenal axis. But when they get their heroin during withdrawal, so they're given saline on a given day instead of their heroin treatment. But when they get their heroin treatment, they return back to what healthy controls show. So basically, heroin is relieving this anxiety and stress-like response. We see the same thing in animals. If you turn animals loose and allow them to have 23 hours access, okay, intravenously to heroin, they end up taking enough heroin that would kill a normal rat on one injection. And if you look at the orange circles. And what's interesting, and we've seen this with all drugs of addiction, as they escalate their intake, their reward thresholds go up. In other words, they're feeling less reward for the same amount of stimulus to their medial forebrain bundle. So they're getting less reward, but they're taking more heroin. Again, it's called hedonic tolerance. Opioids can cause pain as well. Historically, the phenomenon of opioid-induced hyperalgesia has been described as early as the 19th century. In 1870, the physician Albert described the phenomenon, stating, "At such times, I have certainly felt it as a great responsibility to say that pain, which I know is an evil, is less injurious than morphia, which may be also an evil. Does morphia tend to encourage the very pain it pretends to relieve?" And in 1880, Dr. Rosbach also drew a similar conclusion to his predecessor, saying, "When dependence on opioids finally becomes an illness of itself, opposite effects like restlessness, sleep disturbance, hyperalgesia, neuralgia, and irritability become manifest." Even 100 years later, the phenomenon of opioid-induced hyperalgesia continues to be seen by practitioners, 100 years plus, seen by practitioners treating patients who are on chronic opioid treatment. And even in the laboratory, opioid withdrawal-induced hyperalgesia has been demonstrated in heroin-dependent individuals. In these particular individuals, acute withdrawal, of course, produces the most decrease in threshold for pain and increase in decrease in tolerance to pain. But I want you to note is that on the right-hand side, the decreased tolerance to pain is even seen 30 months after stopping opiate use. That's 2.5 years later. Alcohol causes hyperkatifeia and hyperalgesia. So these are data I pulled out of the literature. One's a study by Sandy Brown and Mark Schuckett from 1988. They were looking at clinical depression. This is on the left-hand side, clinical depression in individuals during withdrawal from alcohol dependence. And they're using the Hamilton depression scores. And you can see that the patients almost reach clinical depression levels, not quite the cutoff point you would prescribe an SSRI or antidepressant. And they decline in the next couple of weeks. But if you know the scoring system for the Hamilton Depression Score, you know that they're not exactly tiptoeing through the tulips either at 4 weeks out. And then rooting around in the literature, I actually found another chap who exactly replicated this study in his PhD thesis at the University of Adelaide 11 years later, using the Beck Depression Inventory, exactly the same results. So acute withdrawal, pretty dramatic scores on the BDI, but not really returning to super baseline or below baseline to tiptoeing through the tulips either. Alcohol-induced hyperkatifeia and hyperalgesia is observed in humans and rats. Again, the left-hand side, there are decreased pain thresholds in humans during acute alcohol withdrawal. In a paper by Yoakum et al. In 2010, anxiety elicited by alcohol cues in alcohol use disorder has been demonstrated by Rajita Singha in 2009. We have parallel effects in rodent models, decreased pain thresholds in rats during alcohol withdrawal, increased anxiety-like responses on the elevated plus maze, again during acute withdrawal. So what about the intersection of physical pain and emotional pain? And I hope you'll see where I'm going to be going with all this. So we do congressional briefings from time to time to the congressional staff mostly. Sometimes a House of Representatives congressman will show up, but it's largely for their staff. And we did one on women and alcohol a couple of years ago. And Martha Woodruff, a former public radio journalist and novelist, presented, and she gave such a wonderful talk about alcohol in her life that I asked her for a quote, and this is what she gave me. So I've been using it. Martha passed away just 4 years ago now. "I grew up professionally focused and personally adrift. There's a long history of depression in my family, and my childhood home was dominated by my mother's mental illness that was not recognized, let alone treated. Sometime in my mid-30s, I started using alcohol to provide relief from the pain of childhood damage and depression. Bourbon was the only thing that would make the loop tape of despair in my head pipe down. So how does mood and pain interact with alcohol withdrawal? So subjects undergoing alcohol withdrawal show increased pain sensitivity. That's on the left-hand side. The more severe the withdrawal, the more increase in pain sensitivity. But what's even more interesting is the decreased mood of patient was associated with reduced tolerance to pain. And the higher the depression score was associated with more sensitivity to pain. So again, this was the Beck Depression Inventory, and the more depression, the less tolerant the individual was to pain. This is also shown with opiates, and this is the paper from Koob in 2011, and they showed the unpleasant pictures to individuals who are with opiate use disorder who've been tested on ischemic pain thresholds and tolerance. And what I want you to note is that the individuals during withdrawal who saw the negative pictures had much greater, much less tolerance for pain, and even in the ex-users again, who I believe this was, had stopped taking opiates for quite some time. I don't remember the exact details. But again, we have this in. Hello?
George, can you define tolerance to pain? Because it's different from tolerance to drugs of abuse. And I'm not a pain researcher, and it's the first time I hear about it.
Well, it's how much they can tolerate the pain in this context. It's how much the individual can tolerate the pain.
So, okay. So
how many seconds they can tolerate it. And they can, and here on this graph, you can see it. They can't tolerate it as much, but it's a good question. And thanks for the clarification. So you can see I'm weaving different things together here. So here's another one. And this comes from Jeff Wassenault and his team. Dr. Ferguson put this review together with his team. And they came up with a model of pain as an antecedent to alcohol use, but other drugs included. They call it the CANU model, and it stands for catastrophizing anxiety, negative urgency, and expectancy. And it's an explicit exploration of the role of negative affect in driving alcohol and drug use. Now, obviously, pain can drive alcohol and drug use because you're taking the drug to relieve the pain, right? And that happens a lot, especially with opioids, but it also happens with alcohol. And, drug-related outcomes and expectations have a role in the direct pain relationship. But there's another relationship, which is that the pain causes negative affect, and the negative affect goes on to also perpetuate and drive alcohol, drug use. And in this negative affect context, they talk about pain attitudes like pain catastrophizing, and negative urgency, which can be defined as a disposition to act rashly under duress and undue stress. So in a review article recently, Leandro and I plugged their CANU model into the cycle of addiction. And, while I am focusing on the withdrawal negative affect stage today in this talk, I think the CANU model also invokes protracted withdrawal or protracted hyperkatifeia or whatever term, or protracted abstinence, whatever term you want to use, and moves it into the preoccupation anticipation stage. So, we make these separate parts of the cycle of addiction, but they all blend into one another. And I want to reiterate the point I made earlier, which is you can enter the addiction cycle at any one of these different spots. For example, someone taking opiates for surgery and then continuing the opiates might end up entering through the withdrawal negative affect stage if they can't get off the opiate. So what's the neurobiological basis of hyperkatifeia and hyperalgesia? And every time I do a talk, I find a spelling error, and there's one. Within and between system neuroadaptations. So we're back to the three-stage cycle, and we're going to focus on the extended amygdala for the most part. Before I get to that, we've argued for years that there are both within and between system neuroadaptations associated with hyperkatifeia, opponent process, and negative reinforcement in the withdrawal negative affect stage of the addiction cycle. And this slide just summarizes that. On the left-hand side, you have a loss of the classic reward system for drugs of abuse, drugs of addiction, I should say. And this was a slide I stole years ago from Eric Nestler at Mount Sinai. And you can see that his diagram is darkened here. It used to be in color. So that means you're losing your reward system. But you're also gaining your stress system. And that's illustrated by a blown up version of the extended amygdala and increases in corticotropin releasing factor, norepinephrine, dynorphin, vasopressin, hypocretin, substance P, glucocorticoids, and neuroimmune factors. Today, I'm going to focus on corticotropin releasing factor, glucocorticoids, and dynorphin. And while we haven't done much work on this recently ourselves, there is lying in the background a loss of function in the buffer systems that block stress, that help reduce the stress once it's engaged. And those are NPY, norepeptide Y, nociceptin, endocannabinoids, and oxytocin. So let's look at CRF. CRF was sequenced by Wiley Vale at the Salk Institute in 1981. It is the driver of the brain and body stress response, both via the pituitary and the sympathetic nervous system and in the brain. And all these arrows indicate that CRF has profound effects at different stages of the addiction cycle. I've summarized these arrows in the next slide. So blockade of CRF signaling blocks hyperkatifeia-like responses in alcohol and opioid dependence. So there's plenty of data to support each one of these, except the last one. Alcohol and opioid withdrawal increases extracellular levels of CRF using in vivo microdialysis in the central nucleus of the amygdala of rat. CRF antagonism, systemic, interventricular, or directly into the central nucleus of the amygdala, reduced alcohol and opioid withdrawal behaviors in mice and rats. CRF antagonism, systemic, or in the CEA directly, reduced thermal and mechanical hyperalgesia in alcohol-dependent and heroin-dependent mice and rats. CRF antagonists reduced escalated intravenous opioid self-administration and dependence-induced drinking in mice and rats. CRF antagonists reduced stress-induced reinstatement of alcohol and opioid seeking in rats. However, CRF antagonism failed to reduce alcohol consumption and craving in humans with AUD and in 2 limited human laboratory studies with AUD subjects. And we'll maybe touch on that later. But consistent with this focus on the amygdala, activation in the left amygdala during fearful face processing was altered in heroin-dependent patients after heroin administration, altered during withdrawal and heroin administration. So in a randomized crossover double-blind study, saline and heroin were administered to 22 heroin-dependent patients. The top is healthy controls, the middle is individuals getting saline, and the bottom is individuals getting heroin. Functional magnetic resonance imaging during fearful faces processing in heroin-dependent patients in withdrawal showed a significantly higher left amygdala response in fearful faces than the healthy control subjects. And this higher response was reversed by heroin administration. So on the right-hand side, you can see the increased activation in the amygdala in the patients during withdrawal. And then the bottom trace is going back to what healthy controls show by giving them their heroin. So that's CRF. But Leandro Vendoscolo and I, back when he was a postdoc at Scripps, so dates about almost 15 years now, he kept asking the question, maybe it's not just CRF. What about glucocorticoids? And I kept dismissing his questions because I felt that these were two separate systems acting in two independent ways. So he insisted on doing the study. And I'm showing the prelude to the study in this slide, and then I'll tell you what happened afterward. So the hypothesis is that chronic glucocorticoid activation blocks further activation of the hypothalamic pituitary adrenal axis by blunting CRF mRNA in the paraventricular nucleus. So that's what's shown on the top left-hand corner when Makino, long time ago, gave chronic corticosterone and mini pumps to rats. He completely blunted the expression of CRF mRNA in the paraventricular nucleus. And that's why when you're writing a T32 grant, you don't become cushingoid. So for those of you who don't write grants, the T32 is the worst NIH grant you could write because it's a training grant and it's an enormous amount of work, right? So why don't you get cushingoid? Why don't you? And the reason is because you blunt your PVN and you shut it off during high stress. But what they don't tell you in the textbooks in medical school or graduate school is that in that same time course, you're driving CRF in the amygdala, which is what I was just talking about, corticotropin releasing factor, and that's on the right-hand top panel. Well, we see the same thing with alcohol exposure. There's a blunting of PVN activity with chronic alcohol, but there's a driving of CRF in the amygdala. We see exactly the same thing with opiates. Look at the two bottom panels with Melendez, 2002, McNally and Akil in 2002. They show the same blunting of CRF mRNA in the PVN and driving of CRF in the central nucleus and amygdala. And I might, for those of you who know the history of addiction research, it was actually Mary Jean Creek who originally showed what Melendez showed in that left-hand bottom panel, a blunting of the HPA axis in chronic opiate in individuals with chronic opiate addiction. So Leandro went on to do the study in our laboratory, and it started in 2015. So and a little bit before that, actually, because probably started around 2012. And he showed that alcohol and opiate withdrawal sensitized glucocorticoid receptor signaling in the central nucleus of the amygdala of rats and humans. But here was the one that really blew my hypothesis out of the water, which is that glucocorticoid receptor antagonism, central nucleus, the amygdala, reduced alcohol addiction-like behaviors in both mice, rats, macaques, and humans. And we reviewed that in a paper in 2023. GR antagonism also reduced escalated heroin, sufentanil, methadone, and hydrocodone addiction-like behaviors in rats and zebrafish, even zebrafish. GR antagonism reduced behavioral hyperexcitability and memory deficits caused by alcohol intoxication and withdrawal. GR antagonism reduced thermal and mechanical hyperalgesia in alcohol-dependent and heroin-dependent male and female rats. And GR antagonism reduced alcohol seeking in a human laboratory study published by Leandro in 2015 with Barbara Mason. And an alcohol consumption, and also blocked reduced alcohol consumption, improved mood, sleep quality, and overall clinical status in humans with AUD. Again, a Vendoscolo and Mason collaboration that's unpublished. So more positive results with glucocorticoids in the human condition, leading one to come to the conclusion that came from Alan Schatzberg at Stanford, which is maybe we're putting the fire out in the wrong place. And here's one example of some of our data that we just replicated in a recent study, or Leandro did, showing that a GR antagonist reverses hyperalgesia in alcohol-dependent rats, both in a dose effect dependent manner, both in males and females, using thermal sensitivity. And the final transmitter I'd like to talk about today is the one we're most excited about, and that's dynorphin in the dynorphin kappa system. Again, the same effects of kappa antagonists across the addiction cycle as CRF antagonists and glucocorticoid antagonists. Blockade of the dynorphin kappa signaling blocks hyperkatifeia-like responses in alcohol and opiate dependence. And I'll go through these quickly. Alcohol and opiate withdrawal increases brain levels of dynorphin in the extended amygdala of rats and humans. Kappa receptor antagonism, systemic, interventricular, or in the CEA, reduced alcohol and opioid withdrawal behaviors in mice and rats. Kappa receptor antagonism reduced thermal and mechanical hyperalgesia in alcohol-dependent and heroin-dependent mice and rats. Kappa receptor antagonism reduced escalated intravenous opioid self-administration and dependence-induced drinking in mice and rats. Kappa receptor antagonism reduced stress-induced reinstatement of alcohol and opioid seeking in rats. And kappa receptor antagonism reversed anhedonia and increased reward-related activation in a human clinical trial of subjects with anhedonia and anxiety depression, but was discontinued for lack of efficacy in a phase 3 trial of major depressive disorder, non-SSRI responder subjects. The drug that was used was aticaprant, and I'll talk about that in a minute. A couple of years ago, Renata and Brendan Tunstall and Leandro and I looked at the effects of NorBNI on opioid-induced hyperalgesia, and we saw these dramatic effects. NorBNI has effects that last weeks after one single injection when you give them a very high dose. And I just thought I'd show you these older data because you can see that in the males, some of these effects lasted for 2 weeks after one single injection. In females, it was 7 days with maybe some residual effect at 2 weeks. We just repeated this, and these data are unpublished results from Renata Marchette, Lucy Ward, a postbac, and Courtney Wilkinson, a postdoctoral fellow in our labs. But we looked at dose-dependent reversal of fentanyl-induced hyperalgesia by both a CRF antagonist, R121919, and aticaprant, which is the kappa antagonist that I was speaking about earlier that was given to the human beings. And so this seems to be a pretty robust effect of kappa antagonism and CRF antagonism, in rodents anyway, in reversing opiate-induced hyperalgesia. More recently, Renata Marchette, who's not only a staff scientist and manages my laboratory, but also has a K99. And in her K99 project, she's wanted to look at the dynorphin kappa system in the periaqueductal gray. And so in a mapping study using in situ hybridization with RNA-scope, she was able to show that the predominant expression of prodynorphin and kappa receptors is in the ventrolateral periaqueductal gray, in the green and purple bars. I'm not sure you can see the indications, and you can see some of the fluorescence in the left-hand top corner. And then chemogenetic inhibition of the ventrolateral PAG dynorphin cells had some relatively small effects, but some effects. This is a study that's ongoing by reducing baseline nociceptive responses in females and the protracted withdrawal fentanyl hyperalgesia in males, the 2 circles on the top. And then chemogenetic stimulation of the dynorphin cells increased nociceptive response during acute withdrawal, and that's circled in the bottom. So I want to finish up with a brief discussion of the convergence of pain and negative emotion neurocircuits, the implications for the role of hyperkatifeia in addiction, but also a new pursuit that we're going to be following in the lab at NIDA. So some of you have seen my slide before this. Where do the pain pathways and hyperkatifeia overlap? Well, the spinal thalamic pathway and the spinal parabrachial amygdala pathway all converge on the central nucleus of the amygdala, and you overlap the extended amygdala, and that's where an obvious place for the overlap of pain, physical pain, and emotional pain. And in fact, in preparation for this talk, I decided to look through the literature, and because of a finding we had in the lab, I decided to look through the literature about what is known in the pain field about the spino-parabrachial amygdaloid pathway in the context of chronic pain. And this was a nice diagram of this pathway from Nelson, showing inputs from the spinal cord and the trigeminal nucleus to the parabrachial nucleus, which then goes on directly to the amygdala. And this pathway was long hypothesized by Jean-Marie Besson, who identified it he was working in Paris, and others. Donald Price had hypothesized this. I don't think he had any direct data, but now there's a pretty large amount of data suggesting that this spino-parabrachial amygdaloid pathway may have something to do with emotional pain. So here's some bullet points. The lateral parabrachial nucleus is a major target of nociception specific neurons in the spinal cord and the trigeminal spinal nucleus, as I said. This connection relays various types of primordial sensory information in addition to nociception, such as visceral sense, body temperature and appetite, and avoidance behavior, all contributing to the hypothesized role as one of the brain's alarm signals, this pathway. Unlike the spinal thalamic cortical pathway, somatopathy is limited with the SPA, suggesting that this pathway does not play essential roles in the sensory aspect of pain. The synaptic transmission between the lateral parabrachial nucleus and the central nucleus of the amygdala neurons shows robust synaptic potentiation in various pain models in rodents, suggesting that this pathway is vital to nociplastic aspects of chronic pain. The spinoparabrachial amygdaloid pathway has been implicated in neuropathic pain, chronic pain, and injury-induced pain sensitization. And the CEA is the site for innate and learned fear threat against aversive events and has an essential role in emotional memory, making the spinoparabrachial amygdaloid pathway likely for the emotional aspect of pain. So in a recent study in collaboration with Dr. Maricela Morales from the neuronal network section at NIDA, Maricela is now retired, but also with postdoc Dr. Courtney Wilkinson, Renata I've talked about, Jana Vendroscolo, and Leandro Vendroscolo. We performed an anatomical study to track the dynorphin projections from the CEA using a dynorphin Cree Long Evans rat that we made at NIDA. And what you can see here is there's a representative immunohistochemistry images of the dynorphin neurons in both the CEA and the BNST, and there appear to be cell bodies in the central nucleus of the amygdala, but also a pretty strong projection in both males and females to the bendusis of the stria terminalis. But even more intriguing was what looks to be a pretty big projection of dynorphin terminals to the parabrachial nucleus, the same parabrachial nucleus that I was talking about from the literature in the pain field. So to put all this together, Leandro and I recently put together a review article for Neuron, which is in the final stages of being hopefully accepted. And this diagram comes from that. But there is CRF input to CRF neurons in the bed nucleus stria terminalis. That's been established. I'm now arguing from the data I just showed you that the CEA is projecting dynorphin to the BNST. Dynorphin is also activating the HPA axis, and dynorphin indirectly through a GABA interneuron is activating the CRF system. And I just showed you some data that suggests that there are also dynorphin-containing neurons in the periaqueductal gray that may be contributing to hyperkatifeia. So this is kind of like local circuit, but also a vicious, well, maybe that's too strong a word, but a definite cycle of reciprocal sensitization that appears to be occurring within the extended amygdala. And we're not the only people to come to similar conclusions. Victor Nudgebauer has done some really seminal work on pain modulation in the central nucleus of the amygdala. He argues that nociceptive input reaches the amygdala from the parabrachial nucleus, as we've been discussing, but he argues targeting PKC delta, somatostatin, and CRF neurons. He also argues that endogenous activation of CRF receptors in the central nucleus of the amygdala contributes to aversive, affective, and sensory pain behaviors in different conditions. So, reached the 45-minute time point at my clock. So I'm going to just take 1 minute to tell you what I told you, as I was taught when I was in the Army. Drug addiction represents a dysregulation of incentive salience, pathologic reward, stress, and executive function systems. Pathological habits, reward, stress, and executive function systems corresponding to dysregulation of neurocircuitry with focal points in the basal ganglia, extended amygdala, and prefrontal cortex, respectively. Historically neglected but gaining traction is a focus on the molecular, cellular, and neurocircuitry changes in the brain that drive opponent process, negative reinforcement, and hedonic allostasis. Withdrawal from drugs of addiction cause pain and emotional pain, or hyperalgesia and hyperkatifeia during acute and protracted withdrawal. Neurobiological substrates mediating hyperalgesia and hyperkatifeia include common brain stress neuroadaptations, such as gain of CRF, glucocorticoid, and dynorphin function. Significant overlap in the engagement of brain circuits mediating hyperalgesia and hyperkatifeia may be hypothesized to be mediated by the spinal parabrachial amygdaloid pathway. The hypothesis that dynorphin neurons in the periaqueductal gray and the dynorphin projections to the parabrachial nucleus play a role in the hyperalgesia hyperkatifeia interface is under current investigation. And a focus on brain circuits responsible for hyperalgesia and hyperkatifeia provides a rich substrate for novel approaches to diagnosis, treatment, and prevention of addiction. So this is the lab. We're joined at the hip. We work with each other. Leandro is a Statman Fellow and a colleague with whom I've worked now for my tenure at NIAAA. Jana Vendruscolo is our super biologist who helps with all the studies. Adriana Greg Flores is a lab technician who's on some of the studies I showed you. And I talked about Renato Marchette, our staff scientist. Some of the postdocs who were involved in these studies are Courtney Wilkinson down at the bottom, second from the left on the bottom second row. And some of the postbacs involved are Lucy Ward and Bella Buzzi in some of the more recent work that we're doing, and more recently Maeve Johnston. So thank you very much. It's kind of stayed close to my proper time, and I'm happy to take any questions anybody has.
Well, I'll jump in. Y'all can.
Yeah, go ahead.
Yes. Hi. So why do kappa antagonists not work as well as we want them to? They seem to affect negative emotional states well in humans, but don't touch addiction as well as we want them to. What are your thoughts on that?
Well, we've been able to inject rats with 30 milligrams per kilogram of NorBNI and completely reverse the escalation in heroin intake, cocaine intake, and alcohol intake in animals during acquisition. And we've been able to, in a couple studies, reverse their reacquisition. So I would argue that it's not the target that's the problem, it's probably the formulation. NIDA tried giving JD-TIC, which is another kappa antagonist that's like NorBNI, to human beings, but it had profound cardiovascular side effects, and so they pulled the study immediately. No one's wanted to touch kappa antagonists with a barge pole since in the long-acting version. Aticaprant, we're starting to see pretty good positive effects in our animal models. So, and I know, I was just talking to you today, Tejeda, this last week in a symposium at the National Academy of Medicine, and I know he's pursuing kappa antagonists with future human studies and collaboration. So I'm not sure I think they're doing better than the CRF antagonists. At least they're getting some effects in humans.
Looks like Robert Lustig has raised his hand. You can unmute yourself.
Dr. Koob, nice to talk with you again. Wonderful talk. Question about hyperkatifeia. You focused on the amygdala, appropriately so. I was wondering if the habenula plays a role here as well, and whether serotonin has a modulating effect on the glucocorticoid and CRF effect.
Yeah, serotonin definitely seems to be modulating activity in the brainstem, and I think interacts with the kappa system, if I remember from Charlie Chafkin's work, from Rafe to so I'm trying to remember where he was looking at the kappa interaction, so I'm blanking on it. But so I think there's certainly elements of serotonin that are involved in the stress-like responses. What was the other part?
Well, the specifically.
Oh, the habenula system. Yeah, the habenula is a punishment system
between the habenula and the amygdala.
Yeah, I don't know about the amygdala. The habenula definitely seems to be controlling the VTA,
right,
and contributing to, very possibly contributing to the within system of neuroadaptation, the loss of dopamine function that's associated with acute withdrawal. So that's where I would link the habenula for the moment. I don't know about interactions with the amygdala. I don't remember any, but that may be my ignorance. I'm not keeping track, so who's next? Is it Dorit or Josh? Or
I think I'm next. So thank you very much.
Josh.
Hey, how are you? Thank you very much for the talk. I liked your presentation of the fact that people, even 30 months after drug withdrawal, still seem to have lowered thresholds for pain. I'm wondering if it's possible that those people started out with lower thresholds for pain, in the sense that, and that was reflecting or driving some of their drug take, right? Because I wonder what you think about that.
Very possible. And I don't know that that's been controlled for, although anecdotally, I remember being in a taxi when we used to take taxis, and with a lawyer who found out what I did for a living and said, Dr. Koob, I don't understand why all my patients who have drug problems are always in so much pain.
Yeah.
And presumably they were dried out by the time they got to court. Who knows? At least temporarily.
Okay, thank
Dorit.
you.
I have a general question. So as far as I know, from what I've heard, the women and men have different sensitivity to pain. And you had a couple of slides where women were included in the study, but is there, what you showed, is it generalized or is it different?
That's a really good question, Dorit. And as has been the case, and we rail against this at the Alcohol Institute too, because many of the imaging studies that are done in the alcohol field on males and females include males and females, but they don't even break down the difference in the reporting the results historically. So, um, I'm not asking everybody to power their studies to account for male and female sex differences, but it's nice to break down the results to see if there's a hint of something that could generate a new hypothesis. Now, having said that, Dorit, one thing we did find is that it takes a lot more heroin to get female rats dependent. Not the case with mice, but it is the case with rats. We suspect that.
So they are resistant?
They're resistant. So we have to give more heroin or longer treatment to the female rats. And then the data I showed were just balanced on their level of dependence and hyperalgesia. So
it's not.
I think there are six.
So it's not. Sorry. Go ahead.
Yeah, I'm just saying there are sex differences that do fall out. I worry a lot in the field when people report sex differences if they're not accounting for the baseline of the animals. And because a lot of times the difference is in the baseline, not necessarily in the treatment effect, which is going to require proper dose effect functions and things of that nature. But it's a good question, and we should urge our clinical colleagues when they write papers to at least break out the male and females in them. And I think that's being done more and more, and our preclinical colleagues as well.
Thank you.
John, did you have a question?
Yes. Yes. I was wondering if you could comment about another dimension of the sort of pain addiction phenomenon clinically, and that is in the context of opioid-induced hyperalgesia. Not only is pain exacerbated by withdrawal from the opioid, but can actually be exacerbated by the administration of the opioid. So opioid, classically, as I had opioid-induced hyperalgesia is actually an exacerbation of pain produced by the administration of the opioid.
Yeah. So I'm sure, John, you've read in the old days the work of Francis Colpert. But, he basically argues that there's a sign reversal that occurs. And I think you could evoke, and we've replicated, by the way, we replicated what he did in rodents. We just replicated it in our lab at NIAAA. So we have seen the sign reversal, in which case the animals are showing hyperalgesia even while opiates are on board. And so, he invokes a number of hypotheses. I mean, you can think that opponent process is playing a role, but you can also think of conditioning as playing a role in signaling an opponent process in a seagull-like fashion, shepherd seagull-like fashion.
Mm.
So we've been struggling with some of these issues, and it gets a little, to put it in surf terms, gnarly when you try to figure out what's driving what when pain interacts with opiates. But clearly, opiates can exacerbate pain conditions when they're given chronically. And that, I think everybody accepts at this point. And so part of that is tolerance, because people become tolerant to the opioids. And you don't know whether the pain that persists is because the condition is worse or whether it's exacerbated by the chronic administration of opioids.
Well, clinically, you can see this in the use of fentanyl in the postoperative state with relatively few administrations, 1 or 2 of the fentanyl, and have that switch occur.
That's interesting. I didn't know that. If you have a reference to that, I'd love to see that. Send it on. You can find my email. It's easy, george.koob@nih.gov.
Okay, we'll go try and dig that one out.
I'd love to see the data for that, because it's relevant to what we've been struggling with.
Mark here. I just wanted to, as we're at the top of the hour, there may be one more point here. It looked like the comment is about the focus on the brain, but if you had any thoughts about the role of primary sensory neurons playing in any of this, or in the hyperalgesia or hyperkatifeia.
It's a good question, and I don't know the answer to it in the context of addiction and hyperkatifeia. I wouldn't be surprised if there was some component of that was contributing, but I don't know the answer. Maybe some of the pain individuals know, but there's a lot of work on the spinal parabrachial amygdala pathway in the pain field, but there's almost nothing in the addiction field. There's one paper, and they were stimulating it and showing it suppressed, drug-seeking behavior, but that's probably an aversive effect, a pain-like induction effect, a punishment effect, as we call it in psychology. Dorit, did you have one more quick question? Or you just got your hand up?
Okay, we're just permanently up. Dorit, any final comments before we? Again, we want to thank as a community here, your contribution to this fantastic talk, and we're all wiser for it. Thank you so much. Bye-bye.
Thanks, everybody. It was a real pleasure to be with you again.
Thank you.
Thank you.